Hello everyone, my name is ATHIRA. So today we are going to present our topic. So our topic is Chymosin production using genetically modified microorganisms. So my co-team members are MEGHA and SHIVANI. So let's first discuss about what is chymosin. So chymosin that's actually derived from cows that does not produce using genetically modified microorganisms through biotechnology, mainly focusing on fermentation. So our main focus here is this process involves inserting chymosin gene into microorganisms like bacteria, yeast, or fungi. So that enables these microorganisms to produce the enzyme. Next one is the problem identification. So in problem identification, we are going to discuss about three of the categories. So the first one is ethical concerns. So in ethical concerns, traditionally producing chymosin that involves animal welfare issues. So that's a problem. And the second one is supply challenges. Means it depends on sources which is related to animals. So that produces inconsistent supply. And the third one is environmental impact. So in that, the livestock farming that has significant environmental footprints. Next, we are going to discuss about the biological solutions for this problem. So the first one is consistent supply. So genetically modified microorganisms provides a stable and reliable source of chymosin production. So that's the first one. The second one is ethical production. That means it eliminates the use of animally derived enzymes, thus not harming the animals. And the third one is scalability and efficiency. So in this, the fermentation using genetically modified microorganisms is that allows large-scale production to meet the industrial needs. So in this slide, we are going to talk about the steps of the fermentation and GMO process. So here there are mainly 6 steps. So the first one is gene identification and isolation. The second one is gene insertion for the particular microorganism. And the third one is fermentation process. And the fourth one is enzyme extraction and purification. And our fifth step is quality and safety testing. And the last step is application and cheese production. So here, first we have discussed about introduction about chymosin, and the second one about its problem identification, and the third one was about biological solutions that we can take, and the fourth one is about the steps of the fermentation and GMO process. Thank you.
So now we can discuss how this is produced. So traditionally, chymosin was extracted from the stomachs of the calves, but now through genetic engineering, we can produce it using various organisms like bacteria, yeast, and fungi. Escherichia coli is the basic bacteria that is engineered to produce the chymosin effectively because of due to its rapid growth and high yield. Some of the yeast like Rubomyces latis and Saccharomyces cerevisiae are also used in the production of chymosin, so they offer more controlled production processes. Finally, the fungi like Aspergillus niger and Aspergillus oryzae are also used, as these are used in the soy sauce production. These make them familiar and reliable in the food biotechnology. As we have discussed, fermentation is one of the important steps in the chymosin production. So, some of the key factors that we need to analyze when we are discussing about fermentation are the temperature, pH level, the nutrient availability. So, the fermentation is where these microorganisms are cultivated under the optimal conditions to produce the chymosin effectively. So, the optimal temperature is in between 37 degrees Celsius to the 45 degrees Celsius. So, this ensures the microorganisms are active and productive. Next, the optimum pH level is around 5.8 to 6.5, and this is very crucial for the enzyme's activity and stability. Coming to the nutrient availability, We need to provide a nutrient-rich medium that supplies the carbon, nitrogen, and essential minerals that help for the microorganisms' growth or for their survival. We must have a controlled environment that must be free from contamination, and this environment is very important to maintain a consistency in the chymosin production. So through precision fermentations, we can ensure or optimize these conditions and ensure a stable and scalable production process that meets the industrial demands. So the chymosin that is produced through biotechnology is utilized in the food industry. So our primary target or primary target market is the cheese manufacturers. These producers require a consistent and high quality source of chymosin which the biotechnology provides. So coming to the value proposition, the chymosin produced through genetically modified microorganisms provides several advantages like the ethical production method is increasingly important as consumers demand and which make them more sustainable and animal friendly products. So this chymosin is essentially used in the manufacturing of cheeses like cheddar, gouda and mozzarella. So this GMO implemented production of chymosin not only ensures better quality but also ensures the yield or increases the yield, so making the production process more efficient. So in conclusion, we can say the biotechnology is an revolutionizing the chymosin production, making it more ethical, sustainable and scalable, which in turn supports the ever-growing cheese industry. Next, we will discuss about the overall benefits of the production of chymosin.
So, hello everyone. Now I'm going to talk about the enhancement cheese production. So, first we are talking about the purity and the activity. Biotechnology enhance the purity and the activity of the chymosin. This result in a high quality cheese, a specific type of the cheese. The chymosin is essential in making a popular cheese like a cheddar, Gouda, and the mozzarella. So, moving forward, the safety and the efficiency of the recombinant DNA technology, the biotechnology safety assurance, no live genetically modified microorganism cells are or recombinant DNA are the present into the final product. This ensures that there is a no compromise in the safety, efficiency, precise fermentation enhance the production efficiency by optimizing the yield and the minimizing the cost. Controlled conditions, factors like the temperature, pH, and the nutrient availability are tightly controlled. This condition improve the enzyme production and reduce the variability in a final product. Now talk about the key benefit of the chymosin production. So constant, ethical and the scalable production. Chymosin is produced in a consistent and the scalable manner in using biotechnology. This method reduces dependency on animal sources and addresses the ethical concerns. Improved cheese quality. Biotechnology enhanced the purity and the activity of the chymosin. This leads to a better quality in a cheese such as the cheddar and the mozzarella. Safe and the efficient process. The safety of the biotechnology process is supported by the extensive research. These processes are also highly efficient, optimizing production while minimizing cost. Now talk about the hurdles, the regulatory challenges associated with the GMOs, public perceptions and the acceptance of the genetically modified products that are the hurdles of chymosin productions. Now from the above information we can conclude the significance of the chymosin, the ethical and the environmental impact. Chymosin production through GMM address the ethical concern, ensure a reliable enzyme supply, and tackle the environmental challenges, efficiency and the sustainability. Precise fermentation technology enhances the production's efficiency and promotes the sustainability in the food production. So now talking about the future outlook, biotechnology enhancements, ongoing enhancements in biotechnology are expected to future improve sustainability and the efficiency in a food production. So these are the some reference. You can go through this link for detailed information. Thank you.